Great, so we're going to get started here at the BPI Theater in booth 5476. And we're going to continue with our outsourcing theme, which is part of our larger focus on novel technologies and techniques theme. And our next presenter is Mike Mergis from Catalent. He's the director of Catalent's Biologics Group. And his presentation titled Benefits to Strategic Outsourcing of Biologics Analytical Studies. So I'd like to invite Mike up to the podium.
Thank you for that introduction, Brian. And thank you to Bioprocess International for the invitation to speak here this afternoon. I'm going to speak for a little while on using a case study to explain the benefit to strategic outsourcing of biologics analytical studies. I'm certainly feeling the excitement here at Bio this year. And the feel-good factor goes beyond just the manufacturing of biologics, but to the analytical testing of biologics. And I'm going to spend a few moments going through the market size and the growth of the market, and then use a recent case study of a bioassay development project to explain how Catalent's expertise and capabilities can be brought to bear in these situations. The market itself is segmented about $500 million. The U.S. market is growing, segmented between characterization, method development, stability, raw materials testing, viral clearance, microbiology. And the growth drivers primarily are underlying biologics manufacturing. We have a lot of small companies that are switching from manufacturing their small molecules into biologics, and they have limited expertise with biologics analytical. We have a situation now where there is increasing regulatory requirements and getting ever more complex. The clinical timelines to get analytical testing is becoming faster and faster, and more testing is happening earlier in development. And finally, new specialized technologies are available to bring those clinical timelines forward. When you take a look at the segments to the left, bioassay is the #1 most outsourced service by biomanufacturers and is expected to be the #1 growth assay within the biologics analytical market. Catalent brings over 25 years of experience comprehensively with an orthogonal approach to characterizing and testing biologics. Our centers of excellence cover both GLP and GMP capabilities. We are under one quality system overseeing all of our sites. And most importantly, we have over 100 degreed scientists, with many of those being at the PhD level. Our scientific project leadership team has an average of 18 years of experience in the industry. Our project management and operations leadership team within Biologics Analytical has over 23 years on average experience. We have a very broad range of bioanalytical capabilities. We provide method development at any phase, method optimization, method transfer, whether it be in or out, and we'll be hearing about that a little bit more in the next talk. And we offer phase-specific validation of all of the methods. We perform investigational testing, release testing through commercial, stability testing, in-process testing, and as always seems to be part of a project that I needed tested in a hurry, ad hoc testing. We also provide reference standard characterization and qualifications for all of the different types of projects. I'm going to now take you through a case study of a recent bioassay development project. It was a high throughput assay for virus neutralization. The product is a hyperimmune serum used in immunocompromised patients. The challenge was to rapidly identify plasma donor samples with very high antibody titer with as little as 2 to 3 days of turnaround. The current process was complex, cumbersome, very slow. It was 10 days, low throughput, and the assay itself was very prone to false positives and false negatives. Our new partner was aware of recent literature suggesting that a PCR assay could quantitate the viral replication, and that the neutralizing antibodies should block the infection, depress replication, and this should be capable of being measured in a simpler and faster assay than what they currently had. Our partner had the concept, they knew where they wanted to go, but they did not have the industrial design experience for these types of bioassays. They did not have the flexibility to take on a novel challenge rapidly, and they did not have the specific technical or expertise nor the resources to not only develop the assay, but more importantly, manage a high volume of testing downstream. The insights that were provided collaboratively in discussions with our new research partner built upon that literature base, envisioning an industrial assay process. It focused on fit-for-purpose solutions. We drafted multiple orthogonal approaches to solving the problem, and all the while sought simplicity, speed, rapid proof of concept, and we designed in a high throughput format from the very start. So we're essentially able to transform the raw concept that was brought to the table into a real solution. The quantitative PCR assay itself is comprised of the extraction of RNA from cells, the reverse transcription into DNA through PCR using fluorescent labeled products, and then those products in the assay are able to be very easily quantitated. We very rapidly prototyped the assay using a matrix range finding approach and DOA that was able to allow us to test simultaneously multiple conditions. In the graphic on the left, you see as we input more virus over an increasing serum antibody titer, you get viral depression or suppression of the viral infectivity. It's represented graphically on the right, where you see in green, in the presence of no virus, no antibody, a very high signal-to-noise ratio, and then matrixed effect showing an ever-increasing or increasing depression of the virus infectivity as the antibody titer is increased. So we're able, essentially, in 5 tests, to come forward with a platform that we believed would be rapid and efficient. We took what was a very classic, very elegant way of measuring viral infectivity that had been around for quite some time and developed a simple modern assay that was now only comprised of 5 steps. The older assay was 8 steps, and from the time the assay was put together with the test samples to the time data was available was 10 days. That turnaround time has now been reduced to 3 days. It is higher throughput at 96 well plate format instead of the lower throughput tissue culture dish assay that we started with. The assay used to be prone to false positives and false negatives, but now with this gradient approach, we do not have any false negatives or false positives in the assay. Very importantly, it was a labor-intensive assay. We've managed to really drastically lower the amount of people resources that are applied to the assay. Cost per sample has been reduced from $65 to $31, and then extremely important to our new research partner, we now have the capacity to measure 1,000 samples a week. In the end, we were able to very rapidly show value to our new partner. It was a collaborative process that very rapidly assimilated their expectations, both in the short term to develop an assay quickly that was able to address the long term in having a high throughput assay with a very fast turnaround. The effectiveness of doing this was brought on by the experience of our team. Our research partner now has a very efficient process for measuring these samples and getting results within 3 days so they can maintain contact with their serum plasma donors. And their productivity has increased almost 25-fold from the previous assay that they were utilizing. So I'm going to leave you today with a testimonial from another one of our research partners. I think that very elegantly, from a person that has been in the industry a very long time, appreciates the ability of a research organization such as Catalent to take an assay, transform it into something that is much more rapid, easy to do, takes a lot less resources, less than half the cost, and was brought to bear rapidly and is an approach that can be used for the very long term. Thank you very much.
Mike, I actually have one question.
Sure.
So the assay you've developed for this client, has it been specifically designed for this client, or is this now an assay that Catalent can utilize with other clients?
So this is a platform assay that's been designed specifically for this client, and they have planned already to use it across multiple virus types. So this was essentially the platform developed for a single virus issue that they face, but they plan on making it a platform assay across multiple virus types.
So without getting into the proprietary, was there anything that you learned from this process that you can utilize to create maybe a different assay for other clients?
For sure. I think in taking the matrixed and DOE approach, as we do to solving these types of problems, we can certainly apply that to other infectivity assays and many other cell-based assays as well. It's just not the introduction of the virus into the assay adds another level of complexity where the DOE and the matrix approach was really helpful.
So if I can just broaden it a little bit, assay design and development as it impacts the market today, are you getting more and more demands for more information, or how is the assay portion of this market, the testing, really changing? How is it advanced, and what do you see happening in the near future?
I think what's really changing over the last 10 or more years is the fact that the bioassay used to really not be moved around very much, not transferred. It used to be kept in-house. And not only is it being now more and more outsourced and moved around more than it used to be. It used to be pretty much kept in place. But it's also being utilized much earlier in development processes. So we're at the point now where we have partners coming in very early on in clone selection, asking if they can take a quick look at mechanism of action using a potency assay. So that is truly different than the way these processes worked even 5 or 6 years ago.
So when clients are looking for increased assay help and development, when they're looking to outsource, from your experience, what are some of the capabilities, or what is some of the expertise that they should look for to make sure that they actually partner with the right outsourcing company?
First and foremost, I think it's the people that can be brought to bear, the talent, the amount of experience, the depth and breadth of the team, and depth is very important, I think, in initiating a project and maintaining a project. Secondarily, I think it's folks are looking for cost effectiveness and efficiency, and something of value then that can be tangibly measured and discussed early on, that something that can be measured in the end that you points to get to. And so in this particular case, lower cost, much faster turnaround time, lower input in terms of effort to get work done. And you may not always score all of that, but in this case, it was a really win-win-win situation.
Thank you, Mike.
Sure. Does anybody else have any questions? Okay, thank you.
Great. Thank you very much, Michael.